Hello everyone, welcome to the new session of Fungal Diseases in Crop Plants Part 2. So in this session, we are going to study about barley stripes. So this is caused by a fungus. So this occur in barley plant and smut and bunts disease, loose and covered smuts. So these three different types of fungal diseases in the crop plant we are going to study in this session. Okay, so the first one is the barley stripe. So Helminthosporium gramineum. This is the name of the fungus that causes barley stripe. Okay, so the barley stripe is a systemic disease that affects barley plant but no other cereal crops. So other cereal crops are protected. They are not get any infection with this Helminthosporium gramineum fungi, right? But the barley plant, barley crop get infected with this fungi and they causes stripe-like structures. Okay, the fungus is a seed borne and becomes active during the seed germination. So they are seed borne disease. Seed borne means the seeds of the barley is having this particular fungus. This fungal spore may be there or any mycelium may be there, right? So this is a seed borne disease. So when the seed is sown under the ground, right, it seed germinate, but this seed is infected, so it becomes active during the seed germination, invading the coleoptile and infecting each of the leaves as they develop. So the coleoptile get infected, the leaves which comes out after the germination, that also get infected with this fungus. Barley stripe symptoms provide excellent diagnostic features and the characteristic symptoms appear at the tillering stage. So this barley stripe, you can see the plant and you can tell that this is the symptom. It is clearly visible, right? So this narrow yellow stripes develop the full length of the leaf. So here you can see these yellow narrow strips, right? These are the yellow strips that get developed. So this is due to this fungus. These soon turn reddish brown and then dark brown. So here we can see this is the yellow and later it becomes dark brown in color, right? So this is the symptoms that we can see. This is the diagnostic symptom and that we can see the barley leaves and we can tell that this is a barley stripe disease. The leaf tissue die and dry out. So what happened when the leaf tissue that are infected with this fungi? So what happened? The tissue get die and it become dry out. So causing the leaves to split along the stripe. So suppose this is a leaf, right? And this is the infected part. This is the stripe. So what happened? This is dry and later we can see the leaf is divided into splits into two parts in this way, right? So the plants are dwarf, the plants are stunted in growth, they are quite low in height, that is dwarf, and the leaves turn dark in color, twisted and fray. So sometimes the leaves become twisted like this, right? So twisted and fray, and spike usually fails to have seeds. So the spikes, they do not have seeds formation. The conidiophores appears in the clusters of 2 to 6 and are gray to dark brown in color. So this is the conidia that develops, right? The conidia that develops at the conidiophores. So the conidiophores appear in clusters to 2 to 6, though they appear like this, and on the above part the conidia are present. These are the conidiophores. So this is the way asexual reproduction. This is the conidiophores develop for the asexual reproduction, right? So they appears in the cluster of 2 to 6 and and they are gray to dark brown in color. The basal cell of the conidiophore is enlarged. So this is the basal cell. So this is quite enlarged, and the young conidia are light gray turning dark brown with the age. So here you can see some of the conidia. So this becomes dark brown in color when they get mature, but when they are new conidia are formed, so they are light gray in color. They are straight, cylindrical, slightly tapered, have rounded ends, and typically measure 60 to 90 micrometer into 15 to 18 micrometer with 3 to 6 septa. So they are quite cylindrical. They have septa like this. They have septa. So this is conidia. That born on conidiophore. So this is the peculiarity of this conidia, that they are having the 6 septa. An unusual feature of this species during the sporulation is the frequent development of the secondary conidia from the apices of the primary one. So they give rise to secondary conidia like this. Okay, they give rise to the secondary conidia, and they develop from the apices of the primary conidia only. The perfect stage, that is Pyrenophora gramínea, seldom occurs in nature, and it is of no importance in the disease cycle, but is included here because the name is commonly used by mycologists to identify the fungus. So this is the perfect stage. Perfect stage means this is the organism, this is the fungus in which the sexual reproduction has been reported. So that shows the perfect stage. The pathogen can be kept under the control with the fungicide. So we can have some fungicide and we can control this barley stripe. Okay, we can do one thing, we can go for the seed treatment, right? We can treat the seed with the fungicide. So whatever the fungus, the spores, the hyphae that are present in the seed, that dies off. Okay, and the another thing that we can use the resistant varieties of barley crop. This we can use, right? So if the seeds because this is a seed-borne disease, so seed must be treated with the fungicide before it goes for sowing. Its importance in Europe and North America has diminished considerably in the recent years, though the disease is still common in some areas of South America, Africa, and Asia. Another disease that is second is the Smuts and bunts. 4 genera of Basidiomycetes are included under the general heading of Smuts and bunts. So there are 4 genera that belongs to the Basidiomycetes class, and that causes Smut and bunts disease. So they are pathogenic in small grain, cereals, and many grasses, and produce black spores masses that is called teleospores that partially or completely replace heads, spikelets, or kernels. So here you can see these kernel structures. This is all teleospores are present. We are not having any good quality seed. All the seeds are infected. The spikelets has been infected. The kernel has been infected, and that bears teleospores. Right? So this is the pathogen that occur in small grain cereal, the cereals that bear very small grains, and many grasses as well. They all tend to be seed and or soil-borne pathogens. So basically they are seed-borne and also the soil-borne pathogen. Smut disease, very, very important point. Smut disease caused by Astilago species are usually initiated via floral infection. So this Astelago disease. Astelago species causes this smut disease, and this is with the floral infection. They develop systematically in the host plant during the generation following infection, and the smut spore masses are produced instead of grain. So instead of grain, we will have all these smut. Actually, the grains are filled with the fungal spore, and it is something black charcoal like. So that is why it is called as smut. Right? So instead of grain, we will have all spore masses in that. And that is the fungal spore of Astelago. In contrast, bunt disease caused by Tillatia species, very important. So you have to remember smut disease. Smut is caused by Astelago species, right? And bunt is caused by Tillatia species, right? So you have to remember this. So bunt disease caused by Tillatia species initiate infection at the seedling growth stage of the host plant, develop systematically and produce bunt balls instead of grain in the same generation that infection occurred. Okay, so they are infected in seeds, right? These fungus spore or the fungal hyphae are present in the spore, and they are present in the seedling growth stage of the host plant. So they develop systematically and produce bunt ball instead of grain. So instead of grain, you will get bunt balls, right? As we have seen in the case of a smut, they are having spore masses. So here they will have bunt balls in the same generation that infection occurred. Kernel bunt caused by Neovosia indica is initiated exclusively via floral infection and does not develop systematically and produce spores in the infected kernel. So when the kernels are infected, and this we have seen in the Neovosia indica. So this is also one of the species that causes bunt, that is Neovosia indica. Neovosia indica. So Tillatia species, Neovosia indica, that causes bunt disease. Flag smut, that is caused by Eurocystis species, produces black spore masses in the leaves and the stem of diseased plants, and the infection is initiated at the seedling growth stage of the host plant. So another flag smut, very important. Flag smut is caused by Eurocystis species, and the spore masses we can see in the case of leaves and the stems of diseased plant. Some features that occur in the smut and bunts include, they are caused by obligate parasite. Obligate parasite means they are basically the parasitic fungi. They produce dark spore masses that usually appear at the heading growth stage of the host plant. Okay, so they produce dark spore masses, and that is why they are called as smut and bunt, that usually appear at the heading growth stage of the host plant. And so when the plant is having full growth, then only these dark spores will be appear. Most of the genera have developed distinctive physiological races. Smut and bunts are the injurious disease on the cereal crop in the Eastern Hemisphere. So they are particularly abundant in Near and the Middle East and the Indian subcontinent. So basically they are very injurious because they are causing lots of damage to the crop plant as they get infected to the cereal. So cereal, basically the spikes, the kernels part get infected, and all the seeds are filled with this spores, their spores. Numerous wild relatives of the cereal crop species are indigenous to these areas and serve as an alternative host for the smut and bunt causing pathogens. So there are some alternative host also. So some of the indigenous species of the cereal crop, okay, so they work as an alternative host for these fungus. The use of disease-free seeds, fungicide seed dressings, crop rotation, and the resistant varieties are effective as control measures. So we can control this smut and bunts by treating the seeds with the fungicide, or that is fungicide seed dressing. We can also go for the crop rotation and resistant. Some of the resistant varieties are there for this smut and bunts, so that we can opt. Next is loose and covered smuts. Smuts, sometime it is covered, but sometime it is loose. Astilago species. Loose smuts are among the most easily recognized disease affecting the cereal crop. So in the case of cereal crop, this loose smut is very easy to identify, and it's quite common. The spikelets of the diseased plants are transformed into dark olive brown to black spore masses. So again, the spikelets means when the seed are set up. Okay, so the spikelets, they get infected and they all get filled with the olive brown to black spore masses. The disease becomes obvious only after heading. So after heading means after forming the setup of the seed, then only this disease can be seen, right? So as the head emerges, the spore masses are enclosed in delicate grayish-white membranes that rupture prior to or during harvest, releasing powdery spore masses. So when head emerges means when the spikes emerges, when the kernels setup takes place and there is a setting of seeds. So the spore masses at that particular time, these spore masses get filled up in these seeds, okay, and they are enclosed in the delicate grayish white membrane. So when there is a rupture, that is prior to the harvest time, so they release all these powdery spore masses. Disease spreads either by the floral infection, that is Astilago nuda, or by the embryo infection from these spores that may be carried on the seeds or that are on the soil, that is Astilago nigra and Astilago hordeae. Right? So the floral infection has been seen in Astilago nuda, whereas the embryo infection from the spores that has been seen in the case of means that as seeds are infected, that has been seen in the Astilago nigra and Astilago hordeae. The fungus mycelium invades the growing point of the embryo. So when the fungal mycelium is present in the seed, so they invade the embryo and grows internally with the plant until the floral structures are replaced by the spores. So when there is a emerging of the seedlings, right? So along with the seedling, these hyphae travels through the stem, to the leaves, and also to the floral parts. And when in this floral part seeds are set up, all these spores are released into the spores of the fungus are formed in the seeds. So basically they are destroying the crop, okay, and there is a great loss. In because the seeds, there is no seeds actually. All the seeds are get filled with the spores, so that seeds are of no use. They are closely related forms, or the species are recognized on the basis of the morphological characteristics of the fungi, disease symptoms, and our life cycle. So we are having different forms of smuts, and that is because of the related species, related forms that are recognized on the basis of the morphological characteristics of the fungi. What is the characteristic? What is the morphological characteristics of the fungi, their symptoms and the life cycle? On the basis of that, loose and cover smut has been divided. Loose smut of barley and wheat caused by Astilago nuda. Black loose smut of barley and oat caused by Astilago nigra. Covered smut of barley and oat that is caused by Astilago hodei. Very, very important to learn this. So you must memorize this, because these are the questions that may be asked in the examination, that which of the fungus causes loose smut of barley or wheat, that is Astilago nuda. Which of the fungus that causes black loose smut of barley and oat, Astilago nigra. Covered smut of barley and oat is caused by Astilago hodei. Physiological specialization based on the reaction of the crops and varieties have been established within each of these species, right? So we have seen the physiological specialization in all the different species, right? And hybridization has been reported between some forms with the three groups, which suggest that the forms may be more correctly regarded as the variant rather than the distinct species. So on the basis of their physiological specialization, their morphology, their infection to the particular species, on the basis of this three group has been classified. Barley, oat, wheat and many grasses are attacked by smut of the Astilago species, while triticale and rye are rarely infected. Very important this is. So barley, oat and wheat, they get infected by Astilago, but triticale and rye, they do not get infected by this fungus Astilago. So the incidence of loose and covered smuts has decreased substantially in the North America and Europe as a result of use of more effective seed treatment, fungicides and more resistant varieties. So this loose and covered smut disease was decreased. Where? In North America and Europe. How they got decreased? Because this is again the seed borne. So the seed treatment, fungicides and more resistant varieties has been opted. So with the selection of the resistant varieties, we can have rid of this fungi. And also by treating the seeds with the fungicide, the fungus that are present in the seed, they get die. Astilago and Astilago nuda, Astilago tritsi. So the disease caused by this species occur via floral infection and only barley and wheat are susceptible. So susceptible means they get easily infected. So the loose smut of barley and wheat, right? Astilago nuda, that infect the barley, and Astilago tritsi, that is wheat. The disease caused by this species occur via floral infection, right? One form of Astilago nuda affect barley but not wheat. So this Astilago nuda causes disease in barley, but it do not causes any disease in wheat. In the same way, Astilago tritsi affect wheat but not the barley. Otherwise, these two forms are identical. The heads of the diseased plant tend to emerge slightly earlier than those of the healthy plant. So head means the floral part that emerges slightly early if we compare it with the healthy plants. The membranes enclosing the spores masses are very delicate, rupturing soon after the head emerges. So when the head emerges, the spores that has been set up below the this membrane, they get ruptures and the spores are come out. The spores are exposed. The olive brown teleospores are dispersed by wind's current. Floral infection is initiated by the teleospores landing on the flower. So one infection is there, so it can infect the other flower also. So the teliospores then germinate and produce infection mycelia, which invade the ovaries and eventually the embryos of the developing seeds. The fungus remains dormant until the seed germinates. So once the floral part has been set up, so there is an emergence of this teliospores. Teliospores, when they are present in the seeds, right? So the fungus remain dormant until the seed germinate. They are present in the seed. When these seeds are sowed in the next time, right? So this fungus remain dormant until the seed germinate. The mycelium grows systematically with the seedlings, and as the plant approaches heading, the mycelium penetrate the head tissue and convert them to masses of teliospores. So the teliospores are spherical, finely acuminate, possessing short spines on their surface, are lighter in color on one side and measures 5 to 10 micrometer in diameter. So they are quite round in shape, spherical. Germinating teliospores produce a 4-celled promycelium but no sporidia. So they give rise promycelium, right? So that is 4-celled structure but no sporidia. In barley, loose smut is caused by Astilago nuda is difficult to distinguish from the black loose smut caused by Astilago nigra. Loose smut in barley and the black loose smut, right? So this is, you cannot distinguish because it's having the same symptoms, but both the disease are caused by different fungi. That is, loose smut caused by Astilago nuda and black loose smut is caused by Astilago nigra. Usually there is more on development on plant that is infected by Astilago nigra. On means the seeds part, right? So that the part that sets the seeds, okay, the covering of the seeds. So there is more on development on the plant infected by Astilago nigra. However, the variation in on development among the varieties of barley and wheat limits the usefulness of this characteristic as a diagnostic features. Differences in spore size and degree of accumulate are insufficient to distinguish the species with certainty. Differences in spore color, olive brown in Astilago nuda and dark chocolate brown in Astilago nigra, can be useful for the comparative samples are available. So if we are having both the spores, that is Astilago nuda spores is quite olive brown in color, whereas spores of Astilago nigra, that is dark chocolate brown in color. So on the basis of this spores, if both the spores are present, then we can distinguish. A more reliable means of distinguishing the two species is to observe the germination of the teliospores on water agar. So in a laboratory, that when these teliospores are set up to germinate, right, on water agar, then we can differentiate that which is the species. Astilago nigra produces a sporidia on the germinating promycelium, while Astilago nuda does not produce sporidia. Astilago nuda survives between the crop cycles as a dormant mycelium in the embryo and endosperm of the diseased seed. Fungicide seed treatment used to control seed-borne fungi are ineffective. Hot water treatment that were used to disinfect the seeds have now been suppressed by the systemic fungicide such as carboxine. So earlier we were using the hot water treatment, but now we can use systemic fungicide, that is carboxine. Resistant varieties and good cultural practices have reduced the incidence of this disease. So this is all about this session, right? We will have one more session to complete some of the fungal disease. Till then, thank you very much.